Lecture 4 in Gene Expression. We discussed the players involved in translation, and now we're going to take a look at the process. Similar to transcription, we have 3 stages. One is initiation, elongation, and then termination. And we have different slides upcoming where we can look at this in a little bit more detail, but you can see where the players are, where the RNA is, where the ribosomes are involved. And remember, this is now outside of our nucleus in the endoplasmic reticulum, where the ribosomes are studded on the ER. And then we also have the tRNA molecules and the amino acids that are attached to it. Let's take a look at initiation. In initiation, what we have is the one portion or subunit of the ribosome attaches to the mRNA molecule. When we're looking at bacterial cells, they have something called a ribosomal binding site. And that's a part of your RNA that will attach to your ribosome. Remember, I told you ribosomes are made up of RNA. And so we can have these 2 pieces of RNA, a complementary base pair or hybridize, and that just holds the mRNA in place. The second part of initiation is now the anticodon, or the tRNA comes in with its anticodon to match the start codon, and it also brings in methionine. The last part is that the large subunit will come in and finish this kind of initiation complex. Now, as I mentioned before, we have differences in eukaryotes. Eukaryotes don't use a ribosomal binding sequence. Instead, they use that 5' cap that was added as a part of the RNA processing. Okay, and the start codon might be in a slightly different place too. Don't worry about that. Okay, elongation, the second part of translation. Remember I said that we have these 3 sites, these 3 places, the E, the P, and the A site. So as you saw in initiation, we started out with the P site being filled. And in this case, we've actually moved down from 5' to 3'. We are reading this mRNA molecule. And you can see that we already have some amino acids attached together. And we are now ready to add the next one. So what you see is that the new charged tRNA molecule will come in and sit at the A site. Remember, its anticodon here hybridizes to the codon on the mRNA. And what you can see is that this growing chain that was being held by this tRNA molecule is now passed on to the tRNA molecule that's in the A site. So the baton is being passed. And then what will happen is everything will shift down. This tRNA molecule will move to the exit site. He's not holding on to anything anymore, so he can leave. Whereas this one here moves to the P site. He now is holding this growing chain, and then the incoming tRNA molecule with the next amino acid will sit down in the A site. And this continues to happen as we move down the mRNA molecule from the 5' side and move towards the 3' side. The stopping of all this, the termination of all of this, involves a release factor. When we get to the stop codon, there's no tRNA that matches those 3 stop codons. So instead, a release factor sits there, which causes some changes that lead to everything falling apart, right? The ribosomal subunits fall apart, and the mRNA will maybe go and be translated some again. And the important part is that in the end, I have my polypeptide. Now, there are differences that I've pointed out between the way bacteria do this and the way humans do it. And this just points out how the ribosomes are made up. So these are some bacterial ribosomes and eukaryotic ribosomes. And this shows you the small subunit. And the 30S and the 40S just shows there are different sizes or weights, densities, but you could also see that the proteins even have different numbers. The type of RNA that's used in them are different, and they're different here and here as well. And the reason I mention that, the details are not that important, but what you do want to know is that we can take advantage of the differences between bacterial gene expression, translation in this case, right? So we're looking at translation. We can create antibiotics, and we can prevent production of certain proteins that might be essential for the bacteria. And again, the details of how that's done for each antibiotic is not important, but it explains why we really should understand how bacteria work so that we can understand how to make them not work anymore when it comes to infections. We're going to end this with an explanation of how we move from DNA to RNA to protein, right? So this is our DNA, this is our RNA, and this is our protein. Remember, that's the central dogma, is moving through those three, is understanding that DNA is used to make RNA, and then the RNA is read to make the protein. Remember, we said that you have a double-stranded piece of DNA. We don't really care about this top strand. At least the cell doesn't care about that top strand. It's called the coding or the non-template strand. The one that we care about is the 3' to 5'. Often you'll see that being listed on the bottom, but not always. Remember, the reason that we use 3 to 5 as our template is because we're going to build an mRNA strand moving from a 5' to a 3' direction, because RNA polymerase only knows how to go from 5 to 3'. And when we make that mRNA, we just read the base here, and then we bring in the complement down here. We read the base here, bring in the complement down here. That's how the cell does it. T's match up with A's, but when you have an A in DNA, you're not going to have a T in mRNA. Remember, mRNA only has U. So whenever you would think, hey, I got to put a T in there, make sure you put a U in. Then you're going to read the genetic code that we showed you, that big table, and you're going to translate from the RNA language to the protein language. So each one of these, each one of these codons can be found on that table, and then you put in the appropriate amino acid. So what I'd like you to do at the end of this video is to pause and to take this double-stranded DNA and tell me what the RNA would look like, and then tell me what the protein would look like. Now, of course, this is just an exercise that you're going to do for yourself. You don't have to turn that in, but it's a good way to make sure that you understand how the process works. That is the end of today's video, the end of Chapter 12's video. Thank you very much, and stay safe.